Hi, my name is Ananya Kumaresh, and I'm a sophomore biomedical engineering major here at UTD. While in the Neural Interfaces Laboratory this year, I've had the opportunity to work on investigating methods of using nerve stimulation to restore walking ability under the guidance and direction of Dr. Stuart Cogan and his PhD student Rebecca Frederick. According to the National Center for Biotechnology Information, approximately 35% of stroke patients experience some type of gait dysfunction. This is often caused by a weakness in the calf muscles that control walking motions, and it can often lead to balance issues. In the past, functional electrical stimulation has been used to assist with gait rehabilitation. However, these methods only target one muscle group and have limited precision, as the foot often experiences off-target effects like inversion and eversion. In order to mediate this issue, we believe that gait dysfunction could be improved by stimulating the axons in the sciatic nerve that control muscles in the lower half of the leg. A chronic implant like the wireless floating microelectrode array has the ability to evoke both plantar flexion and dorsiflexion movements at the ankle by activating the appropriate muscles through motor axons within the sciatic nerve. It would therefore allow clinicians to better mitigate any off-target effects of stimulation. The purpose of the first stage of our study was to implant the WFMA in an otherwise completely healthy nerve and then use selective stimulation to determine if it could produce the appropriate plantar flexion and dorsiflexion movements. The WFMA is a wireless device that has 16 electrodes located at the tip of each pin so that it can apply electrical pulses to the nerve. Each electrode pin has alternating lengths so that the electrode can sit at two different depths within the nerve and can thereby control different muscle targets in the lower limb. Rebecca Frederick, the graduate student on the project, surgically implanted the WFMA devices in the left sciatic nerve of 6 Sprague dolly rats, taking care to avoid damaging any surrounding musculature. Selective stimulation began 1 week after implantation, and it was conducted weekly until post-op week 8 and then bi-weekly until week 38. The study was conducted until week 38 to avoid age-related complications, since week 35 marks the animals reaching approximately 1 year of age. Each stimulation session consisted of 2 parts, measuring current thresholds and recording videos of limb motion for offline analysis. To measure current thresholds, the wireless telemetry coil in the device was first positioned over the implanted WFMA, and each of the 16 electrodes were tested individually with pulses at a frequency of 1 Hz. For each electrode, the current was gradually increased until a distinguishable twitch was visible. 2 cameras were also positioned so that the lateral and dorsal surfaces of the rear paw were clearly visible. Stimulation video assessments were then conducted to classify the limb motion associated with each applied stimulus. Movements were classified as plantar flexion or dorsiflexion for the primary movements, and then any secondary limb motions such as toe flexion and extension and lateral rotation and medial rotation were noted as well. The image on the right shows all of the classifications of hind limb motion. Results from current threshold data showed that the electrodes that evoked plantar flexion and dorsiflexion had a fairly constant current threshold value throughout the 38-week study. The average current threshold did not significantly increase and remained at or below 20 microamps for most of the electrodes. There was more variability in the plantar flexion values, which is likely due to the organization of axons within the sciatic nerve. Axons innervating muscles that control plantar flexion are spread out across a larger cross-sectional area, and so the same volume of tissue stimulated by an electrode in that region likely recruits fewer axons than an electrode within the smaller region containing axons that innervate dorsiflexion muscles. Results from the stimulation video assessments showed that each of the 6 implanted devices could consistently recruit plantar flexion and dorsiflexion movements throughout the study period. The percentage of electrodes evoking these motions also remained relatively constant, as shown in the table. We also see that secondary movements were also relatively constant. We observed medial rotation and toe flexion were typically associated with plantar flexion, while lateral rotation and toe extension were typically associated with dorsiflexion, and this makes sense given the anatomy of the foot. Overall, our findings indicate that WFMAs could be safely used as a tool for restoring walking ability and provide a stable interface for rehabilitation therapies in stroke patients. Future studies could explore how the placement of the WFMA within the sciatic nerve, based on the fascicular structure, could produce predictable recruitment of certain types of movements. The next steps for the study are to continue with stimulation video assessments in the animals. After that, the electrical stimulation assessments will be combined with data from sensory and motor function tests to provide a more holistic view of the chronic safety and efficacy of the WFMA as a prosthetic device.